Welcome, everyone. My name is Dion Rossiter, and I am the Executive Director of Science at Cal. We're back for another episode of Chatting in Place, where I interview scientists from across UC Berkeley about what's happening on campus and in their labs during the coronavirus pandemic and the shelter in place. Today, I have with us Dr. Danielle Jorgens, and she is the director of the Electron Microscope laboratory on campus at UC Berkeley. So welcome, Danielle. How are you doing?
It's been an interesting time adjusting to working from home. I'm used to being at the bench all day long, every day. So this has been a difficult transition.
Yes, I'm sure everyone is having their fair share of difficult transitions, but I'm sure for somebody who works on instruments and in a lab and really your job is tied to being there, this must be a hard time for you. So why don't you tell everyone, the viewers and the people watching, what is it you do? Tell us a little bit more about the Electron Microscope laboratory and how really an electron microscope works.
That's a great question. So I'll answer in the context of coronavirus because that's the topical kind of thing that's happening right now, obviously. So at the Electron Microscope Lab, we're a core facility on campus, and we service projects that range from everything from looking at semiconductor material to viruses and mammalian cells and a whole host of different systems with different types of questions that are being asked. We have both scanning electron microscopes and transmission electron microscopes. And the real reason why in a coronavirus context, we would use transmission electron microscopy in our work or scanning EM is because of the resolution that's needed. And so just a very brief definition of resolution, it's very generically the distance that you can resolve between 2 points. So the importance of that is really clear when you think about the size of a virus. A virus in general is going to be between 50 and 100 nanometers in width, and that's assuming it's a sphere, so from the edge of the edge, right? And to remind you, a nanometer is about 1,000th or less of a hair strand. And when you're thinking about what modes you can use, what tools you can use to look at that type of structure of that size, you would basically be constrained to about half of the wavelength of light that you use. So photons that are coming from the sun or from a light bulb all have this kind of range of wavelengths that are associated with that type of illumination. And you can't go lower than that with light microscopy. And so to kind of break that barrier, electron microscopy is used. So electron microscopy uses electrons, which have a much smaller wavelength than the photons of light does. And when you get to half of that wavelength, you're at, atomic scale. So you're looking at atoms. It's very, very small. And so, we can look at viruses very easily and very quickly with electron microscopy. And that's why it's really kind of the gold standard that's used in a lot of the epidemiological studies that are being done right now, where you want to look at the structure of the virus, how is it spreading, what is it getting into, what cells are targeted. And it's really, really, really useful when you're trying to figure out what the culprit of a new emerging disease is. And there's a lab that's part of the NIH, it's the NIAID Rocky Mountains Lab. And they have been the kind of first line researchers for imaging coronavirus. And they did this with Ebola and all kinds of viruses, Zika, you name it. They've done this type of imaging before, where they get sent samples from all over the world and all over the US of patients that have some mysterious disease, and they have to be the virus sleuths and go find this virus. And so they'll take samples from a nasal swab or a biopsy of a tissue and go and hunt for these viruses. And it's really an important tool to use electron microscopy. So I encourage folks to seek out their Flickr feed. They have a really great Flickr feed full of images from the virus sleuthing that they've been doing.
So the images that people are seeing right now in the news and online, those are images taken by a microscope like the ones we have at UC Berkeley?
Correct. And importantly, a lot of them are, kind of glossed over, but I don't want to underemphasize how important it is to use the types of tools that we have at UC Berkeley. And we've got multitude of electron microscopes on campus, which we're really fortunate to have. And we have fantastic scientists that, use them in their science craft really, really well. And so for all of the media images that you've seen of coronavirus, almost every single one of them, except for maybe that really, really beautiful artist illustration, are from electron microscopy. And so just to highlight again, like the reason why we know the structure of the coronavirus and how it gets into cells is from a few different science tools. There's electron microscopy, which can show us the spikes that are on top of or on the outside of the capsid of the coronavirus. There's X-ray crystallography, which can help zone in on particular components like the spike proteins, which are red usually in the illustrations that you see. And those are what are responsible for the viruses to get into human cells as well as animal cells. And then there's other tools like sequencing and proteomics, which can help decipher what the structure theoretically could be from the base structure of the proteins. And so those are kind of the big components that we use in science to figure out structurally the actual shape of the virus and how it's actually using that shape to penetrate cells and be taken up and replicated.
So you can see the virus, and you've already started to talk about how you can look at actually the little pieces, because I've been calling the proteins little spikes, right? But those are proteins that help the virus enter the cell, correct?
Yeah. And
so
they're called spike proteins.
Oh, well.
Very accurate.
Very appropriate.
Yes.
Thank you, scientist. So when you're looking, how do you know where to look, or do you just pick any cell? Really, what does the cell tell you about what's happening, or what does the image of the virus tell you about what's happening in the body?
Yeah, that's a really good question. And it'll vary depending on the disease, but with coronavirus specifically, I think one of the things that you have a question about is, where is it trafficking within the body? Where is it doing the damage that it's going to do in the body? And how extensive is that damage? How much of that is just from the virus, inserting itself into cells and replicating and destroying cells as it goes? And how much of that is because it's causing this huge immune reaction within the body, and then the immune cells are coming in and causing damage? And some of the ways that you would answer those types of questions can only be done with electron microscopy. And so the way that this is usually done is you take a sample from a patient, either through a swab, or sometimes in the cases of a lung inflammation situation where there's this pneumonia that's forming, you can take a biopsy from the tissue that's affected and then send that to a laboratory that can process it in appropriate ways, obviously with safety controls, so it doesn't spread the virus, and put it under the microscope. And that can tell you things from either just purely identifying the virus and kind of the sputum of the lung, or from a swab from the throat or the nasal pathways. And just identifying intact virus tells you a lot about the fact that A, it's present, and B, it's replicating. Because if it's not intact, that means that the body is destroying it or it's not surviving in that environment. The second thing that you can learn from something like a tissue biopsy is when that's processed and you cut these very thin slices of the tissue and you look at them, it lays out not just the virus, but the whole cell. And you can see, is it only getting to the cell surface, or is it trafficking within the cell and accumulating new virus particles within the nucleus of the cell? And there's a lot of information that you can get from those types of images that tell you about the nature of how the infection is unfolding, what's being targeted in the cell, what types of cells are being targeted, and how susceptible the cells are to those viruses. And it's not something that, you can do with 100% accuracy all the time. Sometimes it's really hard to find such a small thing inside a large piece of tissue. But scientists that do this are really, really good at it and know what they're looking for. And so once you've seen a virus, as small as it is, you can start to spot them everywhere. And so in a situation like this, the tissue that would be taken for these initial diagnostic purposes would be kind of surveyed throughout the whole entity of it. And then you go on this hunt for a virus, right? So you're sleuthing around for this virus, trying to find it in this big chunk of tissue. And then when you do, you start asking questions like, where is it at in the cell? Where is it accumulating? How much of the virus do I see? that really, really helps inform a lot of the virus kind of baseline knowledge that we start from when it comes to these epidemiological studies, where you try to figure out, like, okay, well, how bad is this virus? How good is it at getting into cells? The next level that I want to highlight, and something that it's starting to happen now, but it's not something that you can do at the start of something like a pandemic. But scientists really try to jump on this as quickly as you can. You take a virus that's viable and you introduce it into cells, or you take a virus that's been stripped of its genetic material so it can't infect anything beyond just getting into a cell, meaning it can't replicate itself. And you start to do studies about how infectious is this virus? How easily does it get taken into the cell? And that tells you a lot about how quickly this virus will spread. Those studies are also done via electron microscopy, where you want to actually track where the virus particle is being trafficked within the cell. Some of this can be done with light microscopy if you have proper targets, but when you have a novel virus like this, it takes some time to develop those types of markers. And so electron microscopy really becomes the linchpin for all of this type of study.
Great. So that was amazing. And it seems like a lot of what you're talking about is you have the biopsy and you have the cell, and maybe later you're doing experiments on how the virus affects the cell and propagates. So are you now are you the team of researchers at UC Berkeley who are working on the coronavirus, or will your research come a little bit later when, as we learn more and more about what's happening?
So since I'm the director of a core facility on campus, we support whatever COVID research is happening on campus as well as we can. There are some projects that are kind of at this stage getting off the ground right now, where people are starting to build these constructs of viruses that are unable to replicate in a mammalian cell or like a human-like cell. And so in those situations, we would be the source where they would come to look at the thin slices as you look through a cell to figure out where it's getting trafficked. But really, the majority of our work is going to probably come later, because where we'll play a role is when more researchers on campus can go back to work and start exploring the questions that will be answered over the next 5+ years, where it's the question of, okay, well, why was this virus so infectious? Why was this virus so capable of spreading between people? There's really a remarkable ability of this virus to spread from human to human, and that's not a trivial matter when it comes to viruses spreading. The other questions that'll be asked is, what types of cells does this virus target? There's a lot known about the virus uptake. The ACE2 protein, which is on the surface of a lot of cells, particularly your nasal cells, your lung cells, and your bronchial pathway cells, have this molecule, and that's part of how the virus is getting in. But there's a cohort of cases that have been reported on where there's a heart involvement, and it's unclear at this stage how much of that is just from inflammation within the body, and how much of that could possibly be the virus migrating around in the body and being uptaken into the heart tissue. And so there will be questions that need to be ferreted out in the next several years of how penetrable is this virus within the body. And the same goes for the gut involvement. There's been cases of some severe gut involvement where there's, secondary effects like vomiting and other things that happen in the bowels, and how much of that is purely from involvement of the immune cell system, and how much of that is directly from the virus infection. And electron microscopy, again, is one of those tools that can be used to confirm and to very, very, very high resolution, with very high resolution, actually go in and drill down to the molecular nature of how the virus is being trafficked in the cells, and how viable it is in those different types of cells and environments.
That's interesting. I hadn't heard that. So it's could be the vomiting and such could either be a response of the immune system, or the virus can actually be attacking the gut.
Well, there's the question of, how much of the gut is responding to just overall the body's kind of thrown out of proportion immune response, or is it possible that the virus is actually causing some sort of local inflammation through infection in that tissue? It's, yeah. The gut particularly is one of those tissues that's really difficult to get those types of data from because it's big, and there's also a microbiome that has its own natural kind of fauna that lives around, and there's bacteria and all sorts of other kind of microbes that inhabit the gut and help it do its beautiful business. But yeah, it's one of those questions, I think, that in time, in the next few years, this is not going to be an immediate answer situation, but in the next few years, those types of questions will get answered.
Right. And these types of viruses, you learn more and more about the coronavirus and the SARS family of viruses, and that will help you better inform if there is another outbreak of another virus strand, correct?
Exactly. And so when it comes back to electron microscopy, one of the real benefits is using high-end, high-resolution, particularly cryoelectron microscopy. And with cryoelectron microscopy, you can basically freeze the virus in a frozen state and image it with very low dose electrons. And there's a 2017 Nobel Prize awarded in chemistry to the scientists that developed this technology. And one of the beautiful things that's happened in the last 20 years, really 10 years, is we now can visualize electrons directly with specialized camera equipment. And so that helps you really, really drill down to near atomic level information in things like viruses. And so that sort of information now is going to inform not necessarily our response to this particular pandemic in the next month, but that's definitely becoming the basis for vaccine development and will become a basis for any other coronavirus family viruses that come up in the next few years. It's no coincidence that the coronavirus family has produced, other epidemics like SARS and MERS, and it would be short-sighted to think that this will be the only one that turns into a pandemic, right? And so, being prepared to spin kind of on our heels and apply all of our scientific knowledge to the next one as fast as we can, even faster than we are doing now, is going to be a real benefit. And so, building up, I think, recognizing that we need to build up our kind of knowledge base of what these viruses look like and how to best find a vaccine to inhibit or to develop our own antibodies against them is going to, pay dividends next time that we have something like this coming up.
Excellent. Well, I want to wrap up, but I've been asking two questions to all of the people I speak to, and I think it's important to ask you this question too. The first one is if you could explain one thing about the coronavirus that's maybe to clear up a misconception, or that a lot of your family members and friends have been asking you to clarify for them, what would that be? What's your message about the coronavirus that you want to get across?
Yeah, that's a great question, and a big one too. I would say this. I think, I'm going to jump off of what other people across the world have been saying, which is, even if you get through this and you've shut everything down and all of a sudden, the shutdown orders are lifted and it didn't feel like much happened, that's a win. That's actually good. The anticlimactic, not much happens result is actually the winning result. That's what we want to have happen. And I know it's really frustrating and kind of counterintuitive, I think, to our nature to have that be the way that this is going to unfold, where, best case results are happening with no discernible results other than it seems like nothing happened. It's a very strange thing. But I think that's one of the biggest lessons that I think people should take in is how important it is that at the end of the day, the hospitals don't get overrun with patients. And at the end of the day, people are not dying from COVID. That's going to be the win. And unfortunately, I think that's getting lost on a lot of people. And, because it's just not a satisfying answer or concept, it's just a really difficult one to kind of get your head around.
Right.
But that would be the one. Yeah.
Yeah. And that leads into, what are you hopeful for? I'm sure you're hopeful that is the outcome, right? Is there anything else that you're hopeful for as it relates to this pandemic?
I hope that we get through this with that anticlimactic ending. I fear that more of the country is going to turn into what's happening in New York. And, I really hope against hope that we end up coming through this in our area and everywhere else in the country that hasn't been super hard hit yet out of this just thinking, oh, okay, well, that wasn't so bad. That would be great. I really hope that ends up happening. Yeah.
Yeah. Well, thank you so much for joining us. It was very fascinating to talk to you. So I hope that maybe when the country opens up again, we can have you giving a live lecture for folks here at Berkeley.
That would be great. Yeah, I love that.
Well, thank you everyone. To learn more about science at Cal and also to ask our scientists questions. We'll be doing these interviews as we're stuck at home. So if you have a question you'd like answered, visit us at science@cal.berkeley.edu. I wanted to get that right. science@cal.berkeley.edu. So thank you everyone for watching, and thank you again, Dr. Danielle Jorgens.